Perfect. Good morning, everybody. This topic is a little bit unusual because it's about the proof of concept in DME. In particular, it's focused on the aqueous humor liquid biopsy. First of all, I think that everything starts with this sentence, that when tissue is an issue, liquid biopsy is non-issue. The concept of liquid biopsy was born in the field of medical oncology, and actually is a test done on a sample of blood, urine, or other body fluids to look for cancer cells, small pieces of RNA, DNA, or the molecules released by cancer cells. But from 1948 to nowadays, many discoveries have been made. And actually, liquid biopsy is a non-invasive technique that gives us the opportunity to have a real-time detection of a different situation, and also we can easily collect samples in a repeated way and also to perform multiple detection. We have, therefore, considerably comprehensive information regarding a disease, and in many cases is much more sensitive as biomarker than those obtained with a tissue biopsy, and is not limited to the oncology field, their application. Moreover, in the liquid biopsy scenario, new actors in a leading role are emerging. In particular, are really interesting the concept of circulating proteins, because we are moving to a multimodal liquid biopsy to obtain the information regarding the phenotype of a disease. Therefore, we have to analyze the omic cascade and focus on the proteome, because the proteome provides a comprehensive map of the protein's interaction associated with the pathways of the disease and give us the opportunity to identify and monitor biomarker, analyzing the proteins in the body fluids. Because we have different kinds of biomarker, we know very well the imaging biomarker that we can easily obtain with the emerging technologies, OCT, OCTA, but really important are the biochemical one, particularly the circulation biomarker are biomarker obtained by sampling biologic fluids. Therefore, there is a strict correlation between liquid biopsy, proteomics, and biomarker. Because, as we have already said, liquid biopsy is a test done on a fluids to check for molecules released in the body fluids. Proteomics give us the opportunity to identify and monitor biomarkers in a fluid, and a biomarker is a defined characteristic that is measured and evaluated as an indication of a normal or a pathogenic process. But what about retinal disease? Aqueous and vitreous humors are suitable fluids to evaluate biomarkers for retinal disorder, and several studies have shown a full correlation regarding the molecular content, analyzing at the same time aqueous humor and vitreous humor. And the protein's concentration in the aqueous humor is really clinically useful, and this represents a milestone in the liquid biopsy approach in several retinal disorders. So let's focus on the omic cascade in the aqueous humor, particularly in the proteome one. Why? Because if we check in the human protein atlas, we can easily see that the eye is a huge source of proteins, and the retina in particular. How we can easily obtain the aqueous humor? With a sample anterior chamber paracentesis, we can easily obtain about 150 to 200 microliters of aqueous humor. The aqueous humor obtained is stored, is prepared in a very precise way, and then analyzed. Analyzed with the classic gel-based approach, or with the ELISA test, and nowadays also with glass chip protein array. This approach is really important because it gives us the opportunity with a small amount of sample to analyze many, many proteins. But what about DME? When we see in this image and this OCT, we can easily see something about the biomarkers, but this kind of DME are really different. And when we analyze an OCT image, we can easily see the well-known imaging biomarkers obtained with the OCT, the subretinal fluid, the status of the external limiting membrane of the ellipsoid zone, intraretinal cyst, hyperreflective retinal foci, vitreomacular interface, drill, and the thickness of the central part of the retina. But we can easily see that these OCT are a little bit different. The edema is not the same kind of edema. It's a different phenotype. So therefore, we are trying to fill in the gap between the imaging biomarker, the image we obtain, and the phenotype, the pathophysiology of the disease itself. In fact, the OMI study showed that in diabetic macular edema, we have an increase of VEGF, inflammatory biomarkers, not only comparing diabetic eyes with non-diabetic eyes, but also comparing diabetic eyes with the edema to diabetic eyes without edema. Moreover, we have an increase of inflammation-related factors and immune cell-related factors, in particular GFAP and KIR4.1. In fact, our study on DME subject revealed that with a cluster analysis, we can easily find two groups of DME. In the second group in particular, we have an increase of hyperreflective foci, subretinal fluid, drill, vessel density. And this second group also show an increase of inflammatory biomarkers, GFAP, and KIR4.1. The correlation shows that there is a positive and statistically significant correlation between subretinal fluid, drill, hyperreflective foci, and glial and neuroinflammatory omic pattern. And in particular, analyzing DME with and without drill, we found that in DME with drill, we have an increase of inflammatory biomarker and also an increase of both KIR4.1 and GFAP. This is due to the Muller cell activity. In fact, as we have already said, during the pathogenesis of DME, the Muller cells start to produce the GFAP due to their dysregulation. Instead, the astrocyte starts to show an apoptosis, therefore they don't produce GFAP. And also, the Muller cell activation was already proved by our group analyzing patients with and without diabetes, and in particular diabetic retinopathy, and show an increase of both GFAP and aquaporins 4. Moreover, the omics study are really useful regarding the response to the treatment. In fact, corticosteroids seem to reduce inflammatory molecules and also VEGF, and instead, bevacizumab adjusts the VEGF. Also, the response to the treatment with a subtraction micropulsar laser treatment show that after the treatment, both VEGF and inflammatory biomarkers are reduced. Also, the biomarkers of Muller cell activity, GFAP and KIR4.1, are reduced, but we don't find any changes related to RPE-related biomarkers. And in particular, the biomarker of therapeutic success related to the Muller cell activity is GFAP. So in conclusion, we can say that future management of chronic eye disorder are linked to the precision medicine approach. And the aqueous humor sampling gives us the opportunity to directly have a biological sample of the eye, and the aqueous humor liquid biopsy seems to overcome the limitation due to the imaging biomarker and clarify the pathophysiology of DME. Thanks.
Thank you very much. That is an outstanding presentation.
Thank you so much.
Which answered so many questions that we are looking for. So do you have any questions? So again, your conclusion is that Muller cells are major player for diabetic macular edema.
Exactly.
Yes.
And we can easily, with the aqueous humor sample, we can prove this concept because, in fact, we have a correlation between the imaging and also the biological and so the pathophysiological processes which are the base of DME.
This is, again, our impression, because if you compare human retinal endothelial cells with Muller cells, and if you induce both of them, it is only Muller cells that produce huge amount of pro-inflammatory molecules, pro-angiogenic molecules. Thank you very much.
Thank you.
Thank you very much, Julia. Now we move on from a little bit clinical, more clinical side.